go. So we're going to enter a few lectures here in the next, I'd say, 2 weeks where there will be some intended duplication. So we'll hit some things that you've been exposed to in biochem undergrad. We'll hit them again with some clinical. They may be a little convoluted, and so I may hit them again. An example will be tamoxifen for breast cancer, which we'll talk about towards the end of today. So let's start with genes and mutations. You should have 14 objectives listed. I think I added one objective last night or yesterday to next hour's lecture. So if everyone, does anyone not have 14 lecture or objectives on their slide? All right, good. And so as is normally the case, the main objective will be talking about the diseases listed and what type of mutations cause them. Are they point mutations, frame shift? You should know those terms. You should know how they're serious. And you can start combining different things we've learned. So you'll know a little bit about the genetic code. You know single amino acid letters. So you could be able to see a protein sequence and say what was the mutation that caused this different protein sequence. All right. So from Dr. Gardner, you've already gotten the kind of real estate of the genome. And today, we're going to be talking about the gene level largely, okay? So if you want to talk about telomeres offline, we can do that. A lot of times students get interested because there's been a lot of popular press on each one of these about what can make us live forever, right? Is telomerase going to make us live forever? And no, it's not, and why, but it's good, that sort of thing. So all offline. Okay, so genes have been sometimes called the fourth unit of function. Let's see if this is working today. And so you'll see them drawn a few ways, but here I'm showing in colors the exons, okay? And so in this whatever beige is introns, a lot of time the introns will just be a line, okay? And so here's your DNA. You get a primary RNA transcript. It gets spliced together where these beige introns get cut out, and then you have mRNA. That's not the only type of RNA. We'll talk about other ones. But largely in the clinic, and when people talk about RNA, if they don't specify, they're usually talking about mRNA because that codes for proteins. We also have tRNAs that are structural transfer RNAs. We have rRNAs, ribozymes. They're involved in ribozymes. And then we have microRNAs, which you'll actually hear more and more about because those are now starting to be used as therapeutics. So they can have, and we'll get into this, regulatory action on other RNAs. So what's the big difference between RNAs and DNAs? We'll see that, and we'll see how the structure manifests to their function. So we'll have a few moments where I kind of seem to delve off into unimportant stuff, and talk about evolution and how genes went on, but you'll see how that affects de novo mutations and how modules get moved around in our genome. So here is a very high-level conceptualized string of functionality in the genome, okay? And so here are what end up becoming different proteins, and there's different modules that have, for example, this thing called a Kringle domain or a serine protease domain or a growth factor domain. And each of those are oftentimes an exon. And so you have a unit of genetic code that is a functional unit. And so if on an evolutionary time frame that exon gets copied somewhere else, it's not just random, will this ATGC maybe work? It's like how phones are made around the world now. You know, you can plug in a GPS in any device now because it's very cheap to make. We would have an exon that is a GPS module. And so those modules get shifted around. And so you can, a protein can instantly, for example, gain some region that can then bind integrin, and then it would have a totally different function, right? Does that make sense? Not only does that happen in an evolutionary time sense, but depending on the mutation type, it could happen in cancers, right? Oftentimes we're talking about SNPs, single nucleotide polymorphisms in cancers, but we'll talk about multiple ones. So I, despite my closeness to the anatomists, have never gotten them to do this. Every year, the day before this lecture, you have lab, and I want one anatomist to yell out the word dystrophin, but no one's ever done that. And the point was, today when I give that lecture, I'm going to talk to you about this protein that is so long that it takes roughly 24 hours for your body to make it. So from when you heard it then to when you heard it now, that's how long it takes. So it has 79 exons. It represents 0.1% of the genome. Okay, so it is very large. And that's just to give you, I think there's actually two that are slightly, just barely longer than that, but they're not all that long. We have other ones that are super small and compact, and there's a reason for that, and we'll get into it. There are genes that only have one exon, okay, and so don't need intron splicing. And so I don't know if I have it on this slide or another slide. I don't know if I include it on another slide, but histones are an example of very small genes that don't need splicing. Yeah, here, yeah. So why would that maybe be good? I don't think I give it to you on the slide. This is often one of those I ask the engineers. Why would you want any gene? First of all, why would you want any gene to have introns, and why would you want any genes not to have introns? Or is it just random? Could just be random evolution. I've got you for 3 hours, so the quicker you go, the quicker you get out. That's how it works. All right, let's start with the one I think you guys know. Why would you maybe want, go ahead, whichever one you want. That can happen, yep. So that is, right, so we'll have a thing on how many genes we have, but one gene segment based on a start codon could actually make multiple different proteins based on different splicing in different tissues, right? So that goes along with that idea that we're super complex, we need a lot of different things. So that's one reason you want introns. Why would you not want an intron? Let's say you had some protein involved in eating and digestion, and so you eat something. Would it be good for that gene to be similar to dystrophin? No, and why not? Right, because you would eat McDonald's and then you would have the necessary machinery to handle it 24 hours later, okay? So we need a lot of histones, and so they get made very quickly. A lot of times, I use the example of eating because you instantly need stuff. A lot of times when we instantly need stuff, we're not working from the genome. We've pre-made stuff, proteins, and they're set. Insulin's a great example of that. We've compartmentalized it, and then we just release it when we need it. But okay, human factor 8 is another example. Here's, it's 200,000 nucleotides long. 90% of it are introns. There's actually another gene in the intron, which is unique, not always the case. So they can be long and they can actually overlap. Okay, so Dr. Gardner, do you talk to them about transposable elements? Okay, great. So I just reviewed this literature this week to see where we stand. This is daunting to me that this is the case. So long story short, everyone here, has anyone not heard the term junk DNA? Okay, good. Right, so I think I've said it to you. So about maybe 20 years ago, there was, all our genome's junk. Then about 10 years ago, oh no, it's not all junk. We're complex. It's all used for regulation. It's actually somewhere in between. So we have some junk DNA, we have some not. There are these things called transposable elements, which you can think of as viral DNA. There's multiple organisms that has them, but basically it's DNA that codes for machinery that allows that DNA to move around and replicate itself. So it's not necessarily good. In fact, normally it's not good, but it happens because it can happen. So we got some of this genetic material in our genome at some point, multiple times probably, actually definitely, and then that could duplicate itself throughout our genome. And as long as it didn't insert itself into a gene we needed, it didn't matter, right? And so a large portion of our genome, almost half, 40%, is made up of this material. Now, sometimes it can actually do something for us. You could imagine it inserting into a regulatory region and abolishing that, and creating a new gene that is beneficial in some way. So you can't just sequence and find these transposable elements and say they're bad always, because sometimes we've incorporated them, but usually, like on the boards, if that ever came up, they're bad, okay? So there's two types. There's DNA transposons and RNA transposons. The DNA transposons, think of them as cut and paste. So you have DNA that codes for something, and it gets cut out, and that same DNA can then go insert somewhere else. The RNA transposons, you have DNA, it's cut out, reverse transcribed to RNA, and then can insert back into the genetic material. The split between those will be important in a moment. I'll talk to you about that. Okay, and the RNA transposons are sub-split into these things called LTRs and non-LTRs, which I believe I have here, yeah. So the LTR is a long terminal repeat element. And let me see if I have one more slide about this. I do. Okay. So here are some schematics of how this could happen, okay? So you could have this transposable element inserted into an exon, okay? And it could be processed, and then that could be part of your mRNA and become part of your protein, okay? It could be inserted in an intron, and then it kind of gets cut out. It doesn't matter. We'll talk about splicing. It could get inserted in an intron and affect the splicing, right? So all of those are possibilities. It could be inserted upstream and affect transcription factor bindings. So it could not affect the final protein, but it could affect the regulation of that protein, okay? And you can also have non-allelic homologous recombination, where you end up kind of getting a mixture of genomes. I'm not going to test you on that. So here's the breakdown of what we have, and the big take-homes I want you to get is that the LTR type, we have a large portion of them in our genome, 8%, but they're not mobile. So these things, when they cut and paste and go all around, especially the RNA ones, they're really sloppy. I don't know if I gave you that number on how sloppy they are, but no. I forget what the number is, but there's a very high error rate. So when they insert, a lot of times they lose the functionality to re-jump around. So we have 8% of our genome that the whole purpose of it was to move around, and it can't move around now, okay? Then in the non-LTRs, we have autonomous and non-autonomous, meaning do they need other parts of the genome to help move around? Non-autonomous does not need anything. It can do it itself, and non-autonomous needs other things. And the autonomous one is called line 1, is the main one, and it represents 17% of our genome. And about 1 in every 150 births has a novel one of those moved somewhere. So that can cause problems, okay? So when you hear de novo mutation, this can be some of those, okay? And then for the non-autonomous, the main one is this ALU, which represents 10% of our genome. All right. I'd say there's probably a low chance that will be in the boards, but it is 1 in 150 births and 40% of our genome, so I thought it was worth covering. Okay, so let's talk about Duchenne muscular dystrophy. This will be on the boards, very testable. 1 in 3,500 males have this with X-linked. It's an X-linked recessive disease. Again, 2/3 coming from due to mom's genetics and 1/3 de novo. And you're having a decrease in your dystrophin gene, which causes an increase. And this is the start of where I will throw some buzzwords at you and not get into them, because I'm going to get into them later, but I just want to hit a neuron again and again. So that increases this thing, MPT, the mitochondrial permeability transition complex. And you'll hear that again from me And that can be bad. If you open it a little, it's okay, and if you open it a lot, it's bad. And basically think of the mitochondria as the powerhouse of your cell, and if you open up this thing, you're losing all the gradients you've built, and it basically causes the cell to be like, time to die. And so you have necrosis, okay? So interestingly, so dystrophin is involved in the muscle, but when this happens and you lose these cells, it's actually replaced by fat and connective tissue. So people with Duchenne muscular dystrophy can actually look kind of swole, like kind of big muscular, but because it's been replaced with fat and connective tissue. Okay, so why do I bring this up? CRISPR, there are studies going on. You've not gotten CRISPR yet, probably, correct? Right, but you've probably heard about it. So basically, it's like a viral therapy. You're getting genetic material in. Just think of it like that for now. So there are clinical trials ongoing. They just finished one in dogs, and I wanted to kind of show you the efficacy of viral therapy, because to the lay, and not just to you guys, everyone hears these popular press things, and it sounds like we are on the cusp of solving all diseases, right? And when you can see figures of what you're actually doing in the body, to me, it helps you retain how much are we actually fixing. So this was acclaimed as like a huge success. So first of all, they used CRISPR to get the genetic material in. It got into 3 to 90% of muscle cells, so quite a large variance. Now imagine you have some genetic deficiency and someone's telling you we're going to give you viral therapy. If they then said it will get into 3 to 90% of your cells and fix them, you'd be like, that sucks. Okay. And it differs by region, right? So in the heart, it fixed 92%, so much better. And here, they did 4 dogs, I think, and so very small n, but here they injected it into the blood, and here they injected it into the muscle, and you can see there's a difference in delivery. So I mentioned viral therapy to you with cystic fibrosis, which you'll hear again from me, but remember, one of the probably first ways it will be good is that you will inhale the viral therapy and fix your lungs, but you won't fix other tissues potentially because of that, okay? All right, so unfortunately I do have 3 hours today which are blocked well. There's an epigenetics lecture that's kind of a long time from now, 1.5 hours or 1.5 months, and very related to regulation. So there will be some overlap and we'll come back to it. It's not the worst thing because you'll get to hear it a few times, but here's our first go at regulation. So you have your gene here in green. You have something called a Tata box. Basically, where are we going to start? Where should we bind to start making the RNA to become the mRNA to become the protein? And then you have these other regions of DNA that transcription factors can bind and do multiple things, open up, basically shut on or off the gene, right? So transcription factors can do things that bind the DNA can do both. And it's doing it, not that you really care about this, but it's doing this by everything has to touch something to actually do it, right? So the DNA can bend back so all these things can happen together. So these lectures in particular are going to be a little annoying in that they're going to end up having a bunch of acronyms that sound the same as molecular machinery comes in, boom, 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 and all these pieces come together. And the common question I get to know is, what of this alphabet soup do I need to remember? So basically, of course, everything I say is testable, but I'm boxing all the really important stuff, which will end up usually being clinically relevant. Let's see if this works. All right, tell me if this is open. I'll try to do something to make it so. What just happened? I assume it is not open, that poll. Yes or no? It's open? Okay. Next one's open? All right, let me try to fix that. Is this open now? No? All right. God. Okay. Let's do it the old way then. Man. Yep. Okay. Turn, talk to each other. Ten seconds. 3, 2, 1. All right. Pretty good. Wow, it's not going to let me remember that. Okay, I'll talk about A and B. So we'll walk through why these would be. I think you guys get the take home that a 3 base pair deletion is not as bad as a 2, right? Does anyone want to raise their hand and admit they don't understand that? Because you're changing a codon, you're shifting everything, right, as you go. But so if you make a mutation here in A, you could be affecting transcription factors, right? I mean, all of these could be bad. If you make a 3 base pair deletion in B, you are removing 1 amino acid in essence, right? If you are, depending on where you do that, if you take a 2 base pair deletion in C, this is an intron, so it could just get removed. It could affect splicing if it's depending on where it is, but it's less likely to be bad. And if you make a 2 base pair or 1 base pair deletion in an exon, then you're doing a frame shift, and the rest of the amino acid will have different amino acid. Rest of the protein will have different amino acids. And so then E is again a 3 base, and F is a nonsense. So I don't know if people picked F. F could have been there because normally I knew you guys were trained to hear nonsense mutation and horrible, and it normally is. I made it at the very end. So if you chop off 3 amino acids, is it a big deal? It could be. A lot of times the ends can be signaling, but a frame shift mutation early on is definitely worse than chopping off a few. Okay. The answer is D, yeah. Sorry, thank you. I should always say that. All right. So genotype is defined as the actual code of what we have, okay? So that is, when you get genotyped, you actually can read the ATGCs. How many did I answer this? How many genomes do we have in us? Genotypes, not phenotypes. No one's answering. How many genotypes do you have in you? A lot. Give me, okay, a number, roughly. 1 million. How many do you have?
500,000.
All right, one more. How many do you have?
10,000.
10,000. Okay. So, right, there's not a right answer to this, but you're all good. It's not one. So generally it's one. But if you have cancer, that's a different genome. If you talk about your gut biome, then you're in the millions, billion, actually be above billions, right? There's more. I think I've already said this. There are more cells that are not us than us in our body. Okay. But so the big one would be cancer to think about for you guys. So phenotype is how does your genome manifest itself in the world? And so two people with, you can take twin studies of the same genome having different phenotypes because one became a, 3-pack-a-day smoker and the other ran marathons or whatever. But it's not just that, it's, where you live, it's all of that, and we'll get into that. And so I probably harp on this more than most people because to me, treating a patient, you basically need to rank order the efficacy of something. And so there is often times where a drug is prescribed and there is known literature of lifestyle interventions are more effective, cheaper, less side effects than the drug. But the drug is, a prescription's written for that because that's what you can do, and it's sometimes a harder battle to help the lifestyle change. But insurances are starting to fund that in some places where there's incentives to. So in New York, for example, this is a bit of a side, not testable, an insurance company funded buying healthy groceries for people instead of prescribing them something because there was more efficacy in getting fiber and low calorie and all that. All right. So a classic example looking at phenotype is in bacteria. If you have glucose around, then the bacteria will decrease this beta-galactosidase. Whereas if they have lactose, they increase it to be able to use that energy source, right? So we have things like that too. Okay, and this is just a fun one. I'll do it if it works. Is this open? No? Okay, well then forget it. I just people come down. I feel like this is very tribal where they end up coming down. Is something mainly nature or nurture? Is it genotype or phenotype or is it the environment or their genome? And it's a mix, but whatever, you guys can discuss that with yourself. I'm more on the side of the environment mattering compared to most people. But okay, so the importance of things that affect the phenotype differ by differing disease. Some things, right, if you have sickle cell anemia, then it's genetic. If you have cystic fibrosis, then it's genetic. If you have lung necrosis, then that's likely your environment. It could be your environment, right? And so a lot of times you will get, especially as people start to get genotyped, the readout will not be, you should do this, you will definitely have this disease. It will be, you have an increased likelihood. And that, it's very frustrating when you hear those words. I would always urge you as a care provider to give numbers because anything above 0 is classified as an increased risk. And sometimes I've read whole things about this population having an increased risk, and then when you look, it's like a 4% increased risk or something compared to, so you really have to measure that, and we'll get into that. There's actually ways to, these are all fuzzy words. There's things called, have I talked to you about QALYs yet? No? You'll hear about this later, but that stands for quality adjusted life years, and so there are ways to measure how much does this benefit you? How much does it cost? What are the downsides? And things like Medicare end up using that to say, will we fund this drug? And so where do we set a metric as this is a bad genetic thing, or this is a disease, or this is a point mutation? A polymorphism has been set at something that, so a polymorphism is we both have a hemoglobin gene. Maybe I have a different ATGC than him at one position. Is that a mutation that's a problem or something, or is it a polymorphism? Well, if over 1% of the population has it, then it's considered a polymorphism. Why 1%? So the way I always point this out, I'm a big science fiction nerd. If there's an alien world where something has 6 fingers, their polymorphism will not be 1%. It would be 1/12, right? So we chose 1% because we have 10 fingers. And so we have a code, we have numbers of base 10, and so we went to 1%. So it's a little arbitrary, okay? And basically you have to care about, does this SNP, single nucleotide polymorphism, actually cause a problem or not, right? Even if 2% of people had it and it's a problem, then you want to discuss it. Okay. I'm trying to, so far on those evals, I'm a little fast, like I'm a 3.6, so I'm trying to slow down, but I'm filling it up with sides, so I'll try to slow it down and keep it on point. All right, so what types of mutations can you have? You can have, so when we say mutation, I sometimes use that word incorrectly when I'm talking about proteins. Mutations are changes to the DNA. You can have transitions or transversions. So transitions are when you're replacing like with like, okay? So these are not all examples, but a G to an A or a C to a T. Transversions are when you're replacing two things that are unlike. So a pyrimidine with a purine, so G to C or C to A, okay? And here are the structures that, like, of course, you don't need to know them, but I'm just showing you, right, why that matters, right? Because you're changing the hydrogen bonding. So the types of deleterious changes from point mutation, so this is changing a base, are numerous, depending, like I gave you in that visual, right? You should go up to that high level to see if you get it, but you can have a silent mutation. So there's codons that represent the same amino acid, right? And so you can have a mutation where there's pretty much no change. And if you want to hear about potential changes at ivory towers, you can talk to me offline, but really no change. You can have a missense mutation, so you're changing an amino acid. A nonsense mutation, you know, is kind of the worst. You're doing a stop codon, and so you're ruining the rest of the protein. And it depends where those are. So if that's not in the coding region, like I said, you might be affecting a transcription factor binding or a splice site. So I might have given you a similar graph to this, but this is just to point out this on the x-axis here are organisms of increasing kind of complexity. And on the y-axis is non-coding DNA. So DNA that does not code for a protein. And long story short, we are almost all non-coding, right? Obviously the coding stuff's very important, but this is the idea that we're complex and we need regulation, and a lot of that DNA is involved in that. And now you know we have that true junk DNA that's jumped around and doesn't seem to be doing much. Okay. So we're going to go through, I think one of the objectives is name diseases that have each of those mutations, right? So we've talked about sickle cell, so and again, this is there's rare cases where we say you should just learn, like E6V is one of those ones that you should learn, memorize, because so much of the population has it, so know that mutation for the boards. So you're changing a glutamic acid to a valine, so you're changing something that's hydrophilic to something hydrophobic, and it causes all that aggregation. Is that a transversion or a transition? I gave you the DNA here. Decide amongst yourselves. All right, do you guys get it? Transversion, purine to, I spelled that wrong, to pyrimidine, Okay, hemophilia B, so this is a deficiency in factor 9, X-linked recessive. Basically, you're having an ATG in the F9 gene region, but it's in the 5' untranslated region, okay? That changes transcription factor binding, which decreases F9 expression, okay? So another example, but untranslated, still greatly affecting the phenotype. Neurofibromatosis type 1, NF1, it makes the neurofibrinogen protein, and so the take home there is that NF1 blocks RAS, which causes growth, so you'll see RAS up in a lot of tumors. It's very variable in disease, and so the phenotype that you see from that changes a lot. So there's different genetics that cause this. So I think I have a picture. Yeah. So you'll see these cafe au lait spots. So again, when you start seeing these things, think as a patient comes to the clinic, this would be how you could diagnose them, or on the boards, patient presents with, right? The cafe au lait spots are not these big things. They're the kind of discolorations here, okay? And then they also have neurofibromas, these bumps, okay? And they have an increased risk of malignant tumors, along with learning disabilities and hypertension. So it is causing a nonsense mutation. So you're going from C to T, and so you're stopping and you're not making the NF1. And it's about 1 in 4,000 people. I believe we already talked about Tay-Sachs, correct? Remind me? Yep, okay. So lysosomal storage disease, you're decreasing this hexoamidase A, and so you're aggregating things in your lysosome. It presents early, the lysosome breaks open and your neurons die. It can be caused, and so there's a few things that can cause Tay-Sachs, but a point mutation is found that alters the splicing of the mRNA. So this is an example where you have a mutation not in the coding region, but it affects the final protein. And again, the Ashkenazi Jewish population is very predisposed to have the genetics that could cause this, okay? So it's about 100 times the chance of their genotype having that point mutation are 100 times more likely than the general population, 1 in 3,600 instead of 1 in 360,000. Now, interestingly, did I tell you this Louisiana story for Tay-Sachs? No? Okay. So this is an example where you have to know your patient population, because the Ashkenazi Jewish population is very well educated on this matter. And so their risk of having a child that has Tay-Sachs in the last, I forget how long, 40 years or something, has dropped 90%. Okay, and so how is that? I think largely they're getting genetic screens and aborting fetuses that have that, because they're going to die after 3 years. So, that's a whole different question about how you deal with that. Maybe they're also choosing who to marry based on genetics. I don't know about that. But in southern Louisiana, there's something called a founder's effect, where when a kind of a colony first starts off, if it's small enough, the first few people can really matter. And so a couple that started off there had this mutation for Tay-Sachs, but they're not of Ashkenazi Jewish descent. And so now there's a lot of people in southern Louisiana that have this mutation that don't know about it and don't screen for it. And so that population is more at risk of having children than Ashkenazi Jews. Okay. So we'll get into splicing, and there's something called a lariat intermediate, and this mutation in Tay-Sachs is affecting that lariat intermediate. So that's, just hold on that. I did want to point out, when you're looking at a mutation in DNA and then thinking about the effect to the protein, you kind of just have to follow through a little bit. So here, this is the most common variant. It's a 4-base insertion in the hexagene, right? So I talked about a splicing thing, a point mutation, but this is a different one. This is a 4-base insertion. And so if you're following along on top in the normal gene here, you have ATA, which is isoleucine, then you have TCC, which is serine, and then TAT, which is tyrosine. And so the TCC, after the first C, what happened in this population is they've inserted a TATC, okay? So that's 4 amino acids. So you are going to shift things. But the point would be, if you didn't look through kind of a Rolodex of what's happening, or used a, the lookup table of what causes the protein, if on a test, or if you looked at what was the first amino acid to change, this first one does not change, just because TCT also codes for serine, okay? And so then the next one over, ATC, is a new amino acid, isoleucine, okay? And so all of those afterwards, there might be some that happen to line up, but basically they all end up getting off. Okay. So we've talked about kind of small things, point mutations, small insertions. You can also have large deletions. An example of that is what causes red color blindness. Another one is CMT, which I think we talked about. But I think I gave you a rarer example that was related. So in this case, I'm listing most of the genes that are involved. And I don't know if I mentioned this before, but the gene CMT1A, there's 2 of them that are very close to identical, 98% identical. And so when you mutate these or delete large fractions, there's a dosage effect to this. So the phenotype, if I want to use that word, differs by the different types of mutations you have. And so this is related to a myelin gene. So if I'm talking about these 2 genes here, this one, PMP22, you can cause it to be duplicated, and that is referred to as Charcot-Marie-Tooth 1 type, okay? And then you can have a mutation that causes PMP22 to be down via a deletion, okay? So those are both, you can have this gene up and down that both cause differing problems with the myelin sheath and cause differing phenotypes. I kind of walked over that a bit, but yeah. So long story short, all those are involved in the myelin sheath, and when you're deleting large fractions, it can affect, depending on the region, up or down, and both of those can be bad to differing degrees. And we already talked about alpha thalassemia as being large deletions you can delete. I think I already gave you guys this, right? Yes. There's, I'm pretty sure I gave you that one already, that you have these 2 alpha genes and they're copied over to other people. To the progeny. So here's an example, which I think have I run through this with you before? I think I copied this slide to an earlier lecture. I did, right? I'm getting nods? Okay. So this is an example of how you can have large genomic changes, right? So if you have very similar genes, when they cross over, you can get 3, in this example, you had 2 alphas and 2 alphas, but if they line up wrong, you can get 3 alphas or 1 alpha, right? And I walked through the combination of all of those, I believe, in the last time I gave this to you. And so the take home there was, so normally in a normal person you have 4, you have 2 and 2 alpha genes, yes? Really? I thought I copied this over. Okay. Everyone want me to go over this again? Yeah, okay. So you have 2 alpha genes on each strand of DNA, and so normally you have 4. Let me see if I have the table summary slide that I thought I gave you before. Have I not given you this table? Sorry, I thought I gave you this earlier too. Okay, so let me slow down then. Okay, so is it a problem? Well, just like that other disease I was talking about, MCT, you have to worry about, is this one of those where it's an all or nothing or a dosage? And when you have 4 of something, well, let's just talk about alpha thalassemia, it is a dosage. If you have all 4, that's normal, you're fine, 4 alphas. If you have 3 alphas, that's good enough, you're fine. And if you have 2, then it can start being a problem. 1's definitely a problem, and 0's lethal, okay? So it matters not only where the crossover happened, but which one you end up getting, okay? So 3 alpha genes, you're fine. 1, you might, if the crossover happens and you have one alpha gene or three alpha genes, you could be good. You don't know if you're going to get the 3 or the 1. And if you have 1 or less, it's bad. So here I just made you a grid of an example of inheritance. So here we have, let's see, what did I have? I had people that had a deletion, two parents that had the same type of deletion. So they have 1 alpha alpha normal, and then their other allele is 1 alpha with a deletion. Okay, so they have 3 alphas to pass or to potentially pass on. And the same thing's over here. So depending on how that crossover happens, you could end up having a child that has all 4 alphas and is fine, or is like the parents and has 3 and is fine, or has gotten the worst from both parents and has 2, okay? And then they could have problems. So you can't just look at how many alphas you have. You have to also look at where the alphas are. So here's an example where one whole strand of DNA is fine. There's 2 alphas in a row, and the other strand has 2 deletions. There's none. And I made, again, the parents the same. So if you look at this, there's a 25% chance the child would have 4 alphas and be completely normal, right? And there's a 50% chance that they would be like the parents and have 2 alphas and have a bit of a problem. And then there's sorry, I don't know what I said there, 50% chance of that. And then there's a 25% chance it would be lethal. They'd have no alphas, okay? And so if you were genetic counseling someone, you would need to, based on, for me, if I give you an exam question of the boards, you should be able to get a grid like this without, filled in and tell us what are the odds of everything happening, or get a pedigree tree, that sort of thing. But a pedigree tree wouldn't really show you this. This I'm trying to get at, where is the alpha deletion? Are they in cis, or are the deletions in trans on different strands, okay? So if you haven't heard that term before, cis, if something's happening in cis, it means connected. So in the genomic world, something happening in cis means on the same DNA strand, meaning like if you were a polymerase or a molecular machinery moving along, you could get to the other thing. So this is a mutation happening in cis. There's 2 deletions here, whereas there's mutations in trans. And so I ask students, I don't know when we're all genotyped, how people will use this. If Match.com would use this material or something, I don't even know what you would do. Would you be like, well, let's roll the dice at 25%, or, which one's better is open for debate. Okay. This is just another table showing the exact same stuff I showed in a different way. And let me pause and see how much longer I have in this to decide where I want to break. Oops, 2 slides, okay. Okay, this should be active now. So this is just a fun thing now, and then I'll show you a clinical thing on the next slide. Tell me is this active? Yes? Okay, whatever. That's a fun thing, I'll just tell you. Okay, so I was gonna say, when do you think viral therapy will actually start happening? Oh wait, sorry, this is a different question than I thought. Cell types, sorry. Yeah, this was, okay. So the long and short of it is, ta-da, it already happened. Last year was the first FDA-approved genomic therapy. So you'll hear about this, Chimera. It was made by Novartis, so I wanted to kind of run you through where we are now that you saw those cell images, okay? And you kind of see the efficacy when we're kind of looking at cells. So this is super effective, and it's not, injecting easily like they did in the dogs. First of all, it's gonna cost about $500,000, one-time treatment though, right? So this is for leukemia, and if you are less than 25 years old, it was this had this is for, obviously it's better for young people because you're saving them for a long time. There was the first case of basically a child that was cured of cancer. This is like the I Am Legend, right? Someone cured cancer. And so there's not a lot of, I don't know if there's a lot. I mean, there's not a huge population in the US, 3,000 per year maybe. So what do? So this is called CAR therapy. CAR stands for chimeric antigen receptor. And what they're doing is they're taking out the patient's T cells. Okay, this is a reason it's kind of justifiably expensive. They're taking out the T cells, and they are, this graphic down here kind of shows it, they're adding in genomic material to make this chimeric receptor, which basically binds the tumor and an antigen from the tumor. And so you put it back into the patient, and then their T and B lymphocytes attack. And so the T cells you're putting in tell the B cells to proliferate. And so you have large remission after 3 months, okay, over 80%. There is a risk of this thing called cytokine release syndrome. So anytime we're doing stuff with immunology, you have the risk of basically, having huge inflammation. And so with some of these therapies, and this isn't just this one, with some of the viral therapies that are hitting the immune system, you'll see case studies where people basically have this horrible fever and they're close to death and maybe some die. But basically you're jacking up their immune system to fight this cancer off, and it seems like most of the time you can get it to work. So this is the first one that's released. There's a lot more in clinical trials. Again, this was a good example because there is a young population, but this isn't, if I can take one more minute, this isn't easy to, if you talk about quality adjusted life years, this isn't easy to extrapolate to many other things, because for older people that get cancer, it's not, in qualities, it's not likely worthwhile to go through the process to get their immune cell out, to modify it, to put it back in. People are trying to design things that you can just inject and then not have to worry about their getting their stuff out, their cells out. All right, let's take a 10-minute break, and we'll come back at 10 after. Come up with any questions. Yeah.
At all?
The only thing I would say, you haven't gotten this from, basically no. The only thing I would say about butter is it might be good. I have to check what it has. It might be good if that was all you were having to do this good thing called autophagy. Have I talked to you about autophagy yet? No. So long story short, you'll get that from me later. But fasting is good, which doesn't sound like butter. But if you fast for long periods of time, it increases this thing called autophagy, where your cells are kind of getting rid of bad cells and cleaning out. And if you want to keep that going, the only thing that can keep that, the things that hurt that are protein and sugar. So if you need calories from some other way, fat's not a bad one. Okay. But that's like pretty out there. So, but I use butter. It's not,
I'd be pretty much.
Okay, yeah.
Two more. Kangen water?
What?
Kangen? What is like alkaline water that everyone is like using? It's like pH 10 or something, or like 9.
Yeah.
But you're supposed to drink it like 1 glass a day or something like that.
Yeah.
So my friend was just promoting it, and he was in somewhere in Colorado. I'm like, I'm
Yeah.
gonna ask you to teach me.
Yeah. I did a shallow dive into, not that, but into alkaline water, and I could not find any data on that aside from like
Kangen water. So
no, I'm not going to give that my stamp of approval.
And so what do you think about the DNA tests? Like when there's so many companies right now that test your DNA and tell you like your background and stuff like that.
Yeah.
Like is it?
That's a first world Christmas gift. Like
Okay.
the level at which they're doing that, it's usually for kicks. It's
Okay.
people that can afford to do it. Like if you now, it could be useful to find stuff. Like so, even I don't know when I give the lecture, I'll let them talk about this. The main company, 23andMe, I checked last year and they still weren't actually sequencing. They were using a chip that told you what you had of the likely alleles. So even there, you're not finding novel stuff. So it's good for all the really bad genetic stuff we tell you, but most of it is not super useful, and that's partly because of the informatics we don't have. So it could become more useful. Like there are companies that tell you, based on your genome, this type of food you can get more nutrition from, or you have problems metabolizing that. That's the kind of thing that's really useful, but it has to be made super easy to be made actually useful. So,
Just because it gets exponentially expensive to do that, to truly sequence it for each person.
no, it's actually getting exponentially cheaper. So it's under $1,000 now.
Yeah.
The problem is,
But relative to just pre-screening for pre.
oh yes. It's much cheaper to use a chip. Although it used to be much cheaper. It's still cheaper, but not. It's
Okay. It's just I submitted one test for heritage DNA and just to see where my heritage background is. And my friend did 23andMe, and she actually did
How much
gene
was- how much that
therapy.
was?
First was like $300 with like diseases.
That's the
mine
all
was
right, so-
only like mine was only like 120.
Yeah.
So, but I just need to know what's my background, where it's from and stuff. And each one is like uses different analogs, and they do like scan different parts of your, I guess, DNA. Let's say, but again, I was just wondering if you know